welcome back to another AP Biology video on my channel, and today we're going to be going over the cell cycle and mitosis. Right, so in today's video we're going to start off with, why do cells divide? Well, cells divide to increase their surface area to volume ratio. That's what this acronym means, and basically what that does is it allows them to pass signals more easily and perform better in carrying out daily processes. And the reason for this is because there's more space on the cell membrane and on the cell overall for things to go on. So some things we should know before we get into this is that a chromosome, here there's an example of one. This is just one chromatid on a chromosome, but it's still considered one chromosome. There's just a clarification thing, because this does get confusing if you don't understand it. So in this chromosome, it's still one chromosome, but there's two chromatids attached on a centromere. And that's what the centromere is. It's like the thing that attaches the two chromatids. So this is a basic diagram of the cell cycle, and we can see here that the majority of the cell's life is spent in interphase. And what interphase is it consists of three kind of subphases: G1, S, and G2. And you can see it says DNA synthesis on S, we'll get to that later. And then there's the mitotic or M phase. And what that is it's basically just the smaller portion of the cell's life, but the actual duplication and the splitting of the cell happens in that phase, and specifically more in cytokinesis. So the first phase of G1 or of interphase is called G1, right here you can see. And what happens is the cell is basically just recovering from this last time it went through mitosis and the mitotic phase, because it does lose a lot of nutrients and things like that when it goes through it, so it does need to recover. So when it recovers, extra organelles and proteins are produced to kind of make up for the ones that were lost, and the cell grows to, be able to prepare for being split. Now this next phase is the S phase, and this is the DNA replication phase. So in this phase, like I just said, DNA replication and synthesis occurs, and 46 single chromatid chromosomes, so the one purple strand that I showed earlier, become 46 with sister chromatids held together by a centromere. So basically what's happening is the 46 single strand chromatids, an extra chromatid is being added on to each one, and they're held together by a centromere, but they're still considered 46 chromosomes. So it's not like there's 92. That's a common misconception. And so in G2 phase of interphase, the microtubules are created. And what these are is they basically form the mitotic spindle, which is the thing that will allow the chromatids during the mitosis to actually be pulled apart and brought into the middle of the cell. And this is just some extra preparation for mitosis, because that is what follows right after here. As we can see, the first phase of mitosis is here, and that is called prophase. And what prophase is basically the chromosomes condense and the mitotic spindle starts to form. So you can see here in my little diagram, the mitotic spindle is starting to form, pretty small, and the chromosomes are condensing in the middle, and the nucleolus in the cell is gone. So there's no nucleolus here. And just a reminder, these are the condensed chromosomes with their centromeres, and each one of these is a sister chromatid. So next we have metaphase, and in metaphase the chromosomes line up in the middle, and what is going on here is the mitotic spindle is forming a little bit more, and the chromosomes are just lined up in the middle. And what happens after that is anaphase. Now this is the phase where the microtubules and the mitotic spindle actually do what I was saying, and they pull the chromatids to the opposite sides of the cell. And at this point you can kind of have a look of what is actually about to happen. So now what's about to happen is in telophase, the chromosomes begin to decondense. So they kind of go from this little X thing to just these chromatids split apart. The spindle disappears, and then the nuclear membrane and the nucleolus, it reappears. I didn't draw in my diagram here, but just know it reappears. And you can see these dotted lines is me trying to show you that the cell is beginning to show signs of the fact that it's going to split. And in cytokinesis, what happens is the cells are basically split, and what happens is the cleavage, something called a cleavage burrow, basically is like almost a sort of squeezes the cells apart, like a rubber band maybe. That's a better way to analogize it. And what happens here is the cells end up splitting, and the two daughter cells, which is what the two individual cells that are created, they're called daughter cells. they are created with an identical genome or DNA makeup to the original parent cell. And one last thing is that there's checkpoints, and what checkpoints are they basically just checking to see if everything is going correctly in the cell cycle. So the first checkpoint that's very notable is the G1 checkpoint, and what this checkpoint does is it checks for cell size, nutrients, and DNA integrity, which just means the DNA, being created properly and being together. So what this is it's on the first phase of the cell cycle, the phase where it's kind of recovering from its past mitosis, and yeah, it's checking for that. The G2 checkpoint is also checking for DNA integrity, but it's also checking to make sure that DNA was replicated correctly during the S phase of interphase. And the spindle checkpoint is during mitosis, and what happens is the sister chromatids attachment to spindle microtubules are checked. So they're making sure that the microtubules and the mitotic spindle are kind of working correctly and attaching to the correct things. And thank you for watching, and that's going to be it for today's video.